For today's set of notes, we're going to be looking at the structure of DNA and how that structure allows it to be the genetic code for our development and all the functions that our bodies need to do, and then also how that structure allows DNA to replicate or make new copies of itself. So let's go ahead and get started. So DNA, which is called deoxyribonucleic acid, deoxyribonucleic acid is the molecule that contains the instructions required for all the biological processes required for an organism. So it is truly the instruction set for living things. And every single cell in the human body, with the exception of red blood cells, other than red blood cells, every single cell in the human body contains the entire set of DNA, which is about 6 billion base pairs. So later on, you're going to learn about these base pairs and how they match up, and it's 6 billion pairs long. Every cell in our body has all, except for red blood cells, has those 6 billion base pairs. But in different cells, genes can be turned on or off depending on the function of that cell. So for example, in skin cells, there's no need for skin cells to ever digest food. They don't need digestive enzymes. So they turn off in skin cells, they turn off that gene that would produce digestive enzymes. But they might turn on the gene that would grow hair. In stomach cells, they would turn on the gene that produces digestive enzymes because they need to break down food, and the gene that grows hair would be turned off. So they both have those genes because they have the complete set of DNA. They can just turn them on or off. And that process of turning on or off certain genes is called gene regulation, and it's very, very important. Gene regulation. So let's actually look at the parts of DNA, and we'll start by breaking down kind of an individual part and then look at how that folds together in what is the DNA double helix. So in every molecule of DNA, we have a phosphate, and you don't need to know what that is. It's just a part of the backbone. So when we're looking over here, this phosphate is a part of this yellow backbone that runs along on each side of that double helix of DNA. So we have a phosphate, we have a deoxyribose, which is just a sugar molecule. That's a type of sugar. It's deoxyribose. And it really does form a pentagon. It's a 5-sided sugar. That is also a part of the backbone. So together, they create, the deoxyribose and the phosphate create the sugar phosphate backbone to DNA. That's what we call it, the sugar phosphate backbone, and it comes from the deoxyribose and that phosphate group. The most important part in terms of providing the instruction is the actual nucleic acid. And that's represented here by this purple offshoot. The nucleic acid is what we would also call a base. These form the base pairs. So when we look at this molecule of DNA over here, it almost looks like a twisted ladder. And that structure is called a double helix. It's the DNA double helix. And there's 4 different base pairs, and we'll look at that again later. But we have adenine, which is abbreviated A. We have thymine, which is abbreviated T. We have cytosine, which is abbreviated with a C. And we have guanine, which is abbreviated with a G. And the reason why these nucleic acids, these base pairs, are so important is because their structure is unique so that they can only bond with one of the other base pairs. So adenine and thymine can bind together, but adenine cannot bind with cytosine or guanine. Adenine and thymine make a base pair. Likewise, thymine can bind with adenine, but it cannot bind or bond with cytosine or guanine. And then cytosine and guanine form a base pair. So those will always pair together, but they can't pair with either adenine or thymine. And you'll see why that's really important a little bit later in the notes. So those are the base pairs. Adenine pairs with thymine, cytosine pairs with guanine, and then this yellow bar that wraps around is our sugar phosphate backbone of that double helix. So this is what it looks like as pictures. Let's go ahead and write this all out. So again, that phosphate and the deoxyribose, which is that sugar, make up the backbone of the DNA double helix. Those four base pairs are adenine, thymine, that forms a pair together, and then there is cytosine and guanine, and they form a pair together. And a kind of silly way of remembering this that I've seen from other videos is that for adenine and thymine, apples are in the tree. So apples in the tree, A and T. And then for cytosine and guanine, cars in the garage for C and G binding together. So if you can remember apples in the tree, cars in the garage, it'll help you remember the base pairs that form. And then it is the sequence, which just means order. The sequence is the order of things. So the sequence of the base pairs determines what type of protein is going to be made by the cell. And you'll learn about that more in our transcription and translation set of notes. But it's the order that is so absolutely important. And then proteins, so this makes proteins, and proteins are the molecules that actually perform processes in our cells. So if we need to break something down, we would use an enzyme, and that enzyme is a protein. If we need to build something in our cells, we would use a different type of enzyme, and that enzyme will also be a protein. Proteins help move our muscles. They help rebuild structures in our bodies. They are so important, and it's our DNA that allows us to make all the different types of proteins that we need in our body. And then, as I mentioned at the very beginning of the notes, those base pairs also are the reason why we are able to duplicate our DNA or replicate our DNA reliably. So DNA base pairing also allows DNA to make more copies of itself while still maintaining the correct order of those base pairs. So making more copies, we would call this replicate. So when DNA replicates, copies itself, the two sides of the DNA strand first separate. That's the first step. The two strands of that double helix will separate, and then new nucleotides will match up with each strand that creates two new strands of identical DNA. And we're going to draw what that looks like below. So if we started with a DNA molecule, let's just make it really short. It's not going to do 6 billion base pairs. We'll do 6 base pairs. So we'll say A, C, C, G, T, A. That'll be our 6 base pairs. That's one side of the DNA. The other side would have that complementary base pairing. So A bonds with T, C bonds with G, C bonds with G, G bonds with C, T bonds with A bonds with T. So those are the two different sides of our DNA molecule. We can draw the backbone on each side of those as well. So we start here, and then step 1 would be that these two sides separate. So they would just move apart. So I'm going to redraw this now. So they separate, and we would have now further apart A, C, C, G, T, A, T, G, G, C, A, T. So now those have separated, and inside the cytoplasm of the cell, there's just lots of nucleotides that are floating around, and they're going to bond up with the help of some enzymes. So the free-floating nucleotides in the cell, a thymine will find that adenine, a guanine will match with that cytosine. Same here. Then we'll find a cytosine, an adenine, and a thymine will bond up, and then those backbones are still there the whole time. Likewise, for this bottom strand, now that there's space that they've separated, we're going to get an adenine, a cytosine, a guanine, a thymine, and another adenine. And again, those sugar backbones are still there. And when it's all said and done, after they've separated and gone through the entire 6 billion base pairs, we now have 2 new strands of DNA that are identical to what they were before. And the reason why that works is because those base pairs have their complementary bases. Thymine can only bond to adenine, and cytosine can only bond to guanine. So cytosine and guanine, cars in the garage, adenine and thymine. It is rare, but sometimes mistakes do happen in this replication process, and a base pair is added in that's not a match, and that's what we would call a mutation. So when we talk about mutations, you might want to add this word. Mutations in our DNA, they come from errors in DNA replication. There is structures in our body that can repair damaged DNA, but depending on when in our development of the lifespan those mutations happen, they can become permanent. And the mutations can either be good or have no effect at all or be very bad for you. So it's not that mutations are always bad. It depends on the protein that results from that mutation. So hopefully this helps you with the structure of DNA, how DNA can copy itself, and then also how the order of those base pairs will ultimately determine the protein that we make, and we'll go into that last part in more detail in our transcription and translation notes. Thanks for watching.